Right now, as you settle into this quiet moment, something remarkable is happening inside your chest. A muscle no larger than your closed fist is contracting and releasing in a rhythm so steady, so reliable, that you've likely forgotten it's there at all. Yet this small organ has been your most faithful companion since before your first breath, and it will remain so until your very last. Your heart beats roughly 100,000 times each day, circulating 5,000 liters of blood through 60,000 miles of vessels, all without a single conscious command from you. The human heart embodies one of nature's most elegant solutions to a fundamental challenge: how to sustain life through constant circulation. It is both mechanical pump and electrical marvel, both sturdy workhorse and delicate precision instrument. Tonight, we'll explore this extraordinary organ not as medical students cramming for exams, but as curious minds seeking to understand the quiet miracle happening within us right now. Each chamber, each valve, each electrical pathway tells a story of millions of years of evolutionary refinement. As we journey through these anatomical landscapes together, let the steady rhythm of your own heartbeat serve as our guide through chambers that fill and empty, valves that open and close, and vessels that branch like rivers carrying the essence of life itself. Before we begin, I'd love to know where in the world you're listening from tonight, and what time it is where you are. There's something wonderful about knowing we're connected across different places and time zones, all drawn together by curiosity about this incredible organ we all share. Let's get into the video. To understand the heart, we must first appreciate where it calls home. Your heart does not float freely within your chest, nor does it sit precisely in the center as many people imagine. Instead, it occupies a specific neighborhood within your body, surrounded by structures that both protect and support its vital work. The heart resides in what anatomists call the thoracic cavity, that protected space within your rib cage that houses some of your most essential organs. More specifically, your heart lives in a region called the mediastinum, a term that literally means standing in the middle. Think of the mediastinum as a central apartment complex within your chest, bordered on either side by your lungs. It's a surprisingly crowded neighborhood when you consider all the vital structures packed into this space. Your heart shares this area with major blood vessels, your esophagus, your trachea, lymph nodes and nerves, all nestled together in a carefully orchestrated arrangement that allows each structure to perform its function without interfering with the others. The heart's position is beautifully asymmetrical. Rather than sitting dead center in your chest, it leans decidedly to the left, with roughly 2/3 of its mass positioned left of your body's midline. This leftward tilt means that when you place your hand over your heart, you're instinctively reaching toward the left side of your chest. Following an anatomical truth your body seems to know intuitively. The heart's apex, its pointed bottom tip, aims downward and to the left, nestling between the ribs at about the level of your fifth intercostal space. Surrounding your heart is a remarkable protective system called the pericardium, a double-layered sac that serves multiple crucial functions. The outer layer, known as the fibrous pericardium, is tough and inelastic, like a protective tent that prevents your heart from overexpanding and anchors it to surrounding structures. Within this outer shell lies the serous pericardium, which consists of two layers that work together like the most sophisticated lubrication system imaginable. These two serous layers, the parietal and visceral pericardium, create a tiny space between them, filled with pericardial fluid. This fluid acts as nature's perfect lubricant, allowing your heart to beat within its protective sac with virtually no friction. Every single heartbeat involves movement and rotation, and without this pericardial fluid, the constant rubbing would create inflammation and impair your heart's function. It's a reminder that even the most basic biological processes require layers of support and protection we rarely consider. The relationship between your heart and your lungs reveals another marvel of anatomical engineering. Your lungs don't simply sit beside your heart, like neighbors in separate houses. Instead, they wrap around it in a gentle embrace, with each lung molding itself to accommodate the heart's presence. The left lung is actually smaller than the right lung, having developed a special indentation called the cardiac notch to make room for your heart's leftward position. This represents millions of years of evolutionary coordination between these vital organ systems. Your heart's relationship with your ribcage provides both protection and reference points for medical professionals. The ribs create a bony cage that shields your heart from most external trauma, while still allowing for the chest expansion necessary for breathing. The heart sits behind your sternum or breastbone, which acts as a natural shield against frontal impacts. Healthcare providers use the spaces between your ribs, called intercostal spaces, as landmarks to locate your heart and assess its function through listening and examination. Below your heart, the diaphragm forms the floor of the thoracic cavity. This dome-shaped muscle, essential for breathing, maintains an intimate relationship with your heart's function. As the diaphragm moves up and down with each breath, it influences the pressure within your chest cavity, which in turn affects how blood returns to your heart. The coordination between breathing and circulation represents yet another example of how your body's systems work in seamless harmony. The size of your heart might surprise you. While we often think of the heart as large and prominent, it's actually quite modest in its dimensions. An average adult heart measures approximately 12 centimeters long, 9 centimeters wide, and 6 centimeters thick. To put this in perspective, if you make a fist with your dominant hand, you're holding something remarkably close to the size and shape of your own heart. This compact organ weighs between 250 and 350 grams, less than a pound, yet it possesses the strength and endurance to pump blood through your entire circulatory system every minute of your life. Individual variation in heart size and position adds another layer of complexity to this anatomical story. Taller people tend to have hearts positioned more vertically within their chest, while shorter individuals often have hearts that sit more horizontally. Athletes frequently develop larger hearts due to the increased demands of regular exercise, a beneficial adaptation that allows for more efficient pumping. Age, body composition, and even pregnancy can influence heart position and size, demonstrating the dynamic nature of cardiac anatomy throughout life. The heart's location within your body represents far more than mere anatomical coincidence. Every aspect of its position, from its protective pericardial housing to its relationship with surrounding organs, reflects evolutionary pressures that have optimized this organ for its essential role. As you breathe quietly and feel your heart beating steadily in your chest, you're experiencing the result of millions of years of anatomical refinement, housed within a perfectly designed biological neighborhood that supports its every beat. Within your chest beats not one simple pump, but a sophisticated four-chambered system that represents one of evolution's most elegant engineering solutions. Your heart's four chambers work in perfect coordination, each with its own specialized role, yet all united in the single purpose of sustaining life through circulation. To understand how blood flows through your body, we must first explore these remarkable chambers and the unique characteristics that make each one perfectly suited to its task. Let us begin our journey through the heart's chambers, where life-sustaining circulation actually starts. The right atrium. This chamber serves as the body's great collecting station, the place where blood returns after completing its journey through your tissues and organs. Think of the right atrium as a welcoming harbor, where ships return from long voyages, carrying the spent cargo of cellular metabolism, and ready to be renewed. The right atrium receives blood through two major highways: the superior vena cava, which brings blood from your head, neck, arms, and upper chest, and the inferior vena cava, which carries blood from your abdomen, pelvis, and legs. Additionally, the coronary sinus, a smaller but equally important vessel, delivers blood that has nourished the heart muscle itself back to this chamber. The right atrium's walls are relatively thin compared to the heart's other chambers, perfectly suited to its role as a receiving chamber rather than a forceful pumping station. The interior of the right atrium reveals fascinating anatomical features that speak to its developmental history. The smooth posterior wall contrasts with the ridged anterior wall, where small muscular bundles, called pectinate muscles, create a distinctive comb-like pattern. These ridges increase the chamber's surface area and help prevent blood from stagnating in corners and recesses. Near the opening of the coronary sinus, you'll find a small flap of tissue called the Thebesian valve, a remnant from embryonic development that serves as a gentle reminder of the heart's complex formation during our earliest weeks of life. From the right atrium, blood flows into the right ventricle, the heart's gateway to pulmonary circulation. The right ventricle represents the heart's first major pumping chamber, though its job differs significantly from its more famous partner, the left ventricle. The right ventricle's mission is to push blood through the relatively short journey to your lungs, where it will release carbon dioxide and pick up fresh oxygen. The architecture of the right ventricle reflects its specialized function. Its walls are considerably thicker than those of the right atrium, but still much thinner than the left ventricle's walls. This intermediate thickness provides just enough force to push blood through the pulmonary circulation without creating the high pressures needed for systemic circulation. The interior surface of the right ventricle is marked by muscular ridges called trabeculae carinae, which give it a rough, textured appearance and help prevent suction that might impede the chamber's filling and emptying. Within the right ventricle, specialized muscle bundles called papillary muscles extend from the walls connected to the tricuspid valve by delicate but strong fibrous cords called chordae tendineae. These structures work together like guy wires, supporting a tent, preventing the valve from being pushed backward into the right atrium when the ventricle contracts forcefully. Across the heart's central wall, or septum, lies the left atrium, the receiving chamber for freshly oxygenated blood returning from your lungs. The left atrium represents a crucial transition point where blood that has just been refreshed with oxygen prepares for its journey throughout your body. 4 pulmonary veins, typically 2 from each lung, deliver this life-giving blood to the left atrium's smooth-walled chamber. The left atrium's walls are slightly thicker than those of the right atrium, reflecting the higher pressures in the systemic circulation. Its interior is notably smoother than the right atrium, with fewer muscular ridges and a more streamlined appearance that facilitates efficient blood flow. The left atrial appendage, a small ear-shaped projection, serves as an additional storage space during filling, though it can sometimes become a site where blood clots form if the heart's rhythm becomes irregular. The left ventricle stands as the heart's most powerful chamber, the mighty engine that drives blood through the vast network of your systemic circulation. This remarkable chamber must generate enough pressure to push blood through the tiniest capillaries in your toes, the complex filtering networks of your kidneys, and the intricate vessels of your brain. The left ventricle's walls are 3 times thicker than those of the right ventricle, a testament to the tremendous workload this chamber shoulders with every beat. The internal architecture of the left ventricle reflects its role as a high-pressure pump like the right ventricle. It features trabecular carinae and papillary muscles, but these structures are more prominent and robust. 2 major papillary muscles, the anterior and posterior, anchor the mitral valve's chordae tendineae with remarkable precision. The left ventricle's shape is more conical than the right ventricle's, and design that optimizes the mechanical efficiency of contraction and ensures complete emptying with each heartbeat. The walls that separate these chambers, collectively known as the cardiac septa, deserve special attention for their crucial role in maintaining the heart's 2 separate circulatory circuits. The interatrial septum divides the right and left atria, while the much thicker interventricular septum separates the 2 ventricles. These walls ensure that oxygen-poor blood returning from your body stays separate from oxygen-rich blood returning from your lungs, maintaining the efficiency of the circulatory system. The interventricular septum is particularly remarkable for its thickness and complexity. This muscular wall is nearly as thick as the left ventricular free wall, and plays an active role in the heart's pumping action. During contraction, the septum doesn't simply serve as a passive barrier, it contributes significantly to the squeezing action that ejects blood from both ventricles. The septum's blood supply comes from both the right and left coronary artery systems, reflecting its importance to overall cardiac function. The variations in wall thickness among the heart's chambers tell a story of functional adaptation that has been refined over millions of years. The atria, with their thin walls, are perfectly designed for receiving blood at relatively low pressures and gently assisting in ventricular filling. The right ventricle's moderate wall thickness generates just enough pressure for pulmonary circulation, while the left ventricle's substantial muscular walls create the high pressures necessary for systemic circulation. These chamber wall adaptations also explain why different types of heart disease affect chambers differently. The high-pressure left ventricle may develop thicker walls in response to conditions like high blood pressure, while the right ventricle adapts to lung diseases that make pulmonary circulation more difficult. Understanding these adaptive responses helps us appreciate how the heart constantly adjusts to meet our body's changing needs throughout our lives. Each chamber's unique characteristics work together to create a circulatory system of remarkable efficiency and reliability. The right side of your heart handles the pulmonary circuit, moving blood the relatively short distance to your lungs and back, while the left side powers the systemic circuit, sending blood on the long journey through your entire body. This dual circuit design ensures that blood is properly oxygenated before being distributed to your tissues, maximizing the efficiency of oxygen delivery and waste removal. As you rest quietly and feel your heart beating steadily in your chest, remember that each beat represents the coordinated contraction of these 4 specialized chambers, each perfectly adapted to its role in sustaining your life through circulation. Within your heart lies one of nature's most elegant timing systems, a collection of 4 one-way gates that open and close with split-second precision to ensure blood flows in exactly the right direction at exactly the right moment. These heart valves represent biological engineering at its finest. Structures so reliable that they perform their function billions of times throughout your lifetime without a single conscious command from you. Each valve has its own personality, its own specialized role, yet all 4 work together in a symphony of coordination that maintains the forward flow of blood through your circulatory system. The tricuspid valve stands as the guardian between your right atrium and right ventricle, earning its name from the 3 delicate leaflets, or cusps, that form its structure. These cusps are translucent, flexible tissues that move like silk curtains in response to pressure changes within your heart. When your right atrium fills with returning blood, the tricuspid valve remains open, allowing blood to flow freely into the right ventricle below. However, the moment the right ventricle begins to contract, pressure builds rapidly within the chamber, and the tricuspid valve snaps shut with remarkable speed. The tricuspid valve's 3 cusps are named for their positions: the anterior, posterior, and septal leaflets. Each cusp is anchored to the heart by an intricate support system that prevents it from being pushed backward into the atrium during ventricular contraction. Delicate but incredibly strong fibrous cords called chordae tendineae connect each cusp to cone-shaped muscles called papillary muscles that project from the ventricular wall. Think of these structures as the rigging of a sailing ship, with the papillary muscles serving as masts and the chordae tendineae as the lines that keep the sails, or valve cusps, properly positioned even in the strongest winds of ventricular contraction. From the right ventricle, blood must pass through the pulmonary valve to reach your lungs, and this valve operates on an entirely different principle. The pulmonary valve belongs to a category called semilunar valves, named for their half-moon-shaped cusps. Unlike the tricuspid valve, which depends on chordae tendineae for support, the pulmonary valve relies purely on pressure differences to function. 3 pocket-like cusps, the right, left, and anterior cusps, form this valve structure. During right ventricular contraction, the rising pressure forces these cusps open, creating a pathway for blood to flow into the pulmonary arteries. However, as soon as the ventricle begins to relax, the falling pressure within the chamber allows the cusps to fall back together, sealing the valve and preventing any backward flow of blood from the lungs. The design is so elegant that the valve closes completely under its own weight and the gentle pressure of blood trying to flow backward. Across the heart, the mitral valve serves as the bicuspid gatekeeper between your left atrium and left ventricle. This valve features only 2 cusps, the anterior and posterior leaflets, but don't let this apparent simplicity fool you. The mitral valve operates under much higher pressures than the tricuspid valve, as it must withstand the powerful contractions of your left ventricle, the heart's strongest chamber. The mitral valve's cusps are larger and thicker than those of the tricuspid valve, adaptations that allow them to handle the increased mechanical stress of left ventricular function. The anterior leaflet is particularly impressive, stretching like a curtain across a significant portion of the valve opening. The posterior leaflet, while smaller, is no less important, featuring several smaller segments that work together to ensure complete closure. The support system for the mitral valve mirrors that of the tricuspid valve, but with enhanced strength. 2 prominent papillary muscles, the anterolateral and postomedial muscles, anchor the valve through numerous chordae tendineae. These supporting structures must be perfectly coordinated, as any disruption to the papillary muscles or chordae tendineae can lead to valve malfunction and the backward flow of blood into the left atrium. The final valve in this cardiac quartet is the aortic valve, the portal through which blood enters your systemic circulation to nourish every cell in your body. Like the pulmonary valve, the aortic valve is a semilunar valve with 3 half-moon-shaped cusps, the right coronary, left coronary, and non-coronary cusps. These names reflect the relationship between the valve cusps and the openings of the coronary arteries that supply blood to your heart muscle. The aortic valve operates under the highest pressures in your entire cardiovascular system. When your left ventricle contracts, it must generate enough force to open this valve and push blood through the vast network of your systemic circulation. The valve cusps are correspondingly robust, designed to withstand pressures that can exceed 120 millimeters of mercury during peak contraction. One of the most remarkable features of the aortic valve is found in the sinuses of Valsalva, small balloon-like dilations in the aortic root just above each cusp. These sinuses serve multiple functions. They help the valve cusps open fully during systole, create swirling currents that assist in valve closure during diastole, and house the openings to the coronary arteries that supply your heart muscle with oxygenated blood. The timing of valve opening and closing represents one of biology's most precise orchestrations. During the filling phase of the cardiac cycle, called diastole, the tricuspid and mitral valves remain open, while the pulmonary and aortic valves stay tightly closed. This allows the ventricles to fill with blood, while preventing any backward flow from the arteries. As the ventricles begin to contract during systole, the tricuspid and mitral valves snap shut first, followed by the opening of the pulmonary and aortic valves, as pressure builds within the contracting chambers. The sounds you hear when listening to a heartbeat, the familiar lub-dub rhythm, are actually the sounds of these valves closing. The first sound, lub, occurs when the tricuspid and mitral valves close simultaneously at the beginning of ventricular contraction. The second sound, dub, happens when the pulmonary and aortic valves close together, as the ventricles relax and begin to fill again. The coordination required for proper valve function extends beyond simple opening and closing. Each valve must respond to subtle pressure changes, coordinate with the electrical conduction system that controls heart rhythm, and adapt to varying demands placed on the cardiovascular system. During exercise, for example, all 4 valves must operate at higher frequencies while maintaining their precision, ensuring that the increased cardiac output reaches your working muscles efficiently. The durability of heart valves astounds even cardiologists who study them professionally. These structures open and close roughly 37 million times per year, experiencing mechanical stresses that would quickly destroy most man-made materials. Yet they perform this function reliably throughout an entire human lifetime, a testament to the remarkable engineering principles embedded in biological design. As your heart beats quietly in your chest right now, these 4 valves are working in perfect harmony, opening and closing in response to pressure changes, ensuring that every drop of blood flows in the right direction at precisely the right moment. Their coordinated function represents one of the most reliable mechanical systems in the natural world, a valvular symphony that sustains your life with every beat. Your heart is built from a type of muscle found nowhere else in your body, a specialized tissue that combines the best features of voluntary and involuntary muscle, while adding unique characteristics that make continuous, rhythmic contraction possible throughout your entire lifetime. Cardiac muscle represents one of evolution's most remarkable innovations, a tissue that must be strong enough to generate significant pressure, coordinated enough to contract in perfect unison, and tireless enough to work without rest for decades. To appreciate the uniqueness of cardiac muscle, we must first understand how it differs from the other 2 types of muscle in your body. Skeletal muscle, the tissue that moves your arms and legs, operates under conscious control and can generate tremendous force, but it tires quickly and requires periods of rest. Smooth muscle, found in your intestines and blood vessels, works involuntarily and can sustain contractions for long periods, but it contracts slowly and generates relatively little force. Cardiac muscle ingeniously combines the desirable traits of both, while eliminating their limitations. Like skeletal muscle, cardiac muscle appears striated under the microscope, revealing the organized bands of contractile proteins that generate force. This striped appearance reflects the highly organized arrangement of actin and myosin filaments within structures called sarcomeres, the fundamental units of muscle contraction. However, unlike skeletal muscle fibers, which can be enormous single cells stretching the entire length of a muscle, cardiac muscle cells are much smaller, typically measuring only 50 to 100 micrometers in length and 10 to 20 micrometers in width. What makes cardiac muscle truly extraordinary lies in how these individual cells connect to one another. Cardiac muscle cells, called cardiomyocytes, are joined by specialized junctions called intercalated discs, structures that appear as dark lines running perpendicular to the muscle fibers when viewed under a microscope. These intercalated discs represent one of the most sophisticated cell-to-cell communication systems in the human body, serving both mechanical and electrical functions that are essential for coordinated heart contraction. The mechanical aspect of intercalated discs involves structures called desmosomes and adherence junctions, which physically link adjacent cardiac cells together with incredible strength. These connections ensure that when one cardiac muscle cell contracts, its force is efficiently transmitted to neighboring cells, allowing the entire heart muscle to work as a unified structure. Without these strong mechanical connections, the force generated by individual cells would be wasted, and coordinated contraction would be impossible. Even more remarkable, are the electrical connections within intercalated discs, specialized channels called gap junctions that allow electrical current to flow directly from one cardiac cell to another. These gap junctions create what physiologists call an electrical syncytium, meaning that the entire heart muscle behaves electrically as if it were one enormous cell. When an electrical impulse reaches one cardiac muscle cell, it can instantly spread to all adjacent cells through these gap junctions, ensuring that the entire heart contracts in a coordinated wave. Within each cardiac muscle cell lies the contractile machinery that generates the force of each heartbeat. Myofibrils, long protein structures that run the length of each cell, contain the actin and myosin filaments responsible for muscle contraction. These filaments are arranged in repeating units called sarcomers, which give cardiac muscle its striated appearance and represent the basic contractile units of the heart. The process of cardiac muscle contraction follows the same sliding filament mechanism found in other striated muscles. Myosin filaments, thick proteins with projecting heads, interact with thinner actin filaments in a process powered by cellular energy in the form of adenosine triphosphate. When calcium ions are released within the cell, they bind to regulatory proteins associated with actin, allowing myosin heads to attach and pull the actin filaments past them, shortening the sarcomere and generating force. What sets cardiac muscle apart is its extraordinary ability to contract rhythmically without external stimulation. While skeletal muscle requires nerve signals to contract and smooth muscle often responds to hormonal or local chemical signals, cardiac muscle possesses an intrinsic ability to generate its own electrical impulses and contract spontaneously. This property, called automaticity, ensures that your heart continues beating even when disconnected from the nervous system, a crucial safety feature that has kept transplanted hearts beating during the surgical procedure. The cellular structure of cardiac muscle reflects the enormous energy demands of continuous contraction. Cardiac muscle cells are packed with mitochondria, the cellular powerhouses that produce the energy needed for muscle contraction. These mitochondria comprise nearly 40% of each cardiac muscle cell's volume, a proportion far higher than found in most other cell types. This mitochondrial density reflects the heart's absolute dependence on aerobic metabolism and explains why the heart muscle is so vulnerable to interruptions in oxygen supply. Unlike skeletal muscle, which can function temporarily without oxygen by switching to less efficient anaerobic metabolism, cardiac muscle has virtually no capacity for anaerobic energy production. This metabolic specialization makes the heart incredibly efficient during normal conditions, but also means that any interruption in oxygen delivery can quickly lead to cell death and heart muscle damage. The heart muscle's blood supply represents another unique aspect of cardiac anatomy. The heart cannot simply absorb oxygen and nutrients from the blood flowing through its chambers. Instead, it requires its own dedicated circulation system called the coronary circulation. This system consists of specialized arteries that branch directly from the aorta, just as blood leaves the heart, ensuring that the heart muscle receives oxygenated blood at the highest pressure available in the circulatory system. The coronary arteries penetrate deep into the heart muscle, branching repeatedly to form an extensive network of smaller vessels that supply every cardiac muscle cell. This rich vascular supply is essential because cardiac muscle cells have virtually no capacity to survive without oxygen, unlike some other tissues that can tolerate brief periods of oxygen deprivation. The timing of coronary blood flow reveals another fascinating adaptation of cardiac muscle. During the powerful contraction phase of the cardiac cycle, the squeezing of the heart muscle actually compresses the coronary arteries, temporarily reducing blood flow to the heart muscle itself. However, during the relaxation phase, when the heart muscle relaxes and the pressure within the ventricles drops, coronary blood flow increases dramatically, allowing the heart muscle to receive the oxygen and nutrients it needs for the next contraction. The cardiac muscle cells themselves display remarkable longevity compared to other cell types in your body. While many cells throughout your body are regularly replaced through cell division, most cardiac muscle cells are formed before birth and must last your entire lifetime. This longevity is both a strength and a vulnerability. It allows the heart to maintain consistent function over decades, but it also means that damage to cardiac muscle cells is often permanent, as they have very limited ability to regenerate. As your heart beats quietly in your chest right now, billions of cardiac muscle cells are working in perfect coordination, each one contributing its small amount of force to the coordinated contraction that propels blood through your circulatory system. These specialized cells represent millions of years of evolutionary refinement, creating a muscle tissue capable of the sustained, coordinated and powerful contractions necessary to maintain life through circulation. Within your heart lies an electrical system so sophisticated that it would make any engineer envious. A network of specialized cells that generate and conduct electrical impulses with precision that rivals the finest electronic circuits. This cardiac electrical system operates independently of your conscious mind, creating the rhythmic impulses that coordinate every heartbeat from your first moment of life until your last. Unlike the electrical systems in your home or car, which require external power sources, your heart generates its own electricity through the remarkable properties of specialized cardiac cells. The story of each heartbeat begins in a small cluster of cells, nestled in the wall of your right atrium, near the opening of the superior vena cava. This tiny region, no larger than a grain of rice, is called the sinoatrial node, though cardiologists often refer to it simply as the SA node. These unassuming cells serve as your heart's natural pacemaker, spontaneously generating electrical impulses that will spread throughout your entire heart and trigger each coordinated contraction. The cells within the sinoatrial node possess a unique property called automaticity, the ability to spontaneously depolarize and generate electrical impulses without any external stimulation. Unlike ordinary cardiac muscle cells, which remain electrically stable until stimulated, pacemaker cells in the SA node slowly leak sodium and calcium ions across their cell membranes, gradually building up electrical charge until they reach a threshold that triggers an action potential. This process repeats continuously, typically 60 to 100 times per minute in a resting adult. What makes the sinoatrial node the heart's master pacemaker is not just its ability to generate electrical impulses, but the speed at which it does so. While other regions of the heart also possess automaticity, the SA node fires more rapidly than any other cardiac tissue. This fastest firing rate ensures that SA node impulses reach and stimulate other potentially automatic tissues before they have a chance to generate their own impulses, maintaining the heart's unified rhythm under normal conditions. From the sinoatrial node, electrical impulses spread rapidly across both atria through specialized conducting pathways and ordinary atrial muscle tissue. This wave of electrical activity travels at roughly 1 meter per second, causing both atria to contract in a coordinated fashion that pushes blood into the ventricles below. However, the electrical impulse cannot simply continue directly into the ventricles due to a crucial anatomical barrier. Between your heart's atria and ventricles lies a band of fibrous tissue called the cardiac skeleton, which serves multiple important functions. This fibrous barrier provides structural support for the heart valves and creates electrical insulation between the atria and ventricles. This insulation is essential because it forces all electrical activity from the atria to funnel through a single specialized pathway where timing can be precisely controlled. The electrical relay station for this carefully controlled conduction is the atrioventricular node, or AV node, located in the lower portion of the right atrium near the tricuspid valve. The AV node serves as both a gateway and a delay station, allowing atrioelectrical impulses to pass through to the ventricles while imposing a crucial timing delay. This delay, typically lasting 1/10 to 2/10 of a second, ensures that the atria have sufficient time to empty their blood into the ventricles before ventricular contraction begins. The cells within the atrioventricular node conduct electrical impulses much more slowly than ordinary cardiac muscle, creating this beneficial delay through their specialized properties. AV node cells have fewer gap junctions connecting them to their neighbors, and these gap junctions contain different proteins that conduct electricity less efficiently. Additionally, AV node cells have fewer sodium channels in their membranes, further slowing electrical conduction through this critical region. From the atrioventricular node, electrical impulses enter the bundle of His, named after the German cardiologist Wilhelm His, Jr., who first described this structure in detail. The bundle of His represents the beginning of the ventricular conduction system, a specialized network of cells designed to rapidly distribute electrical impulses throughout both ventricles. This bundle travels through the fibrous cardiac skeleton via a small opening, representing the only normal electrical connection between the atria and ventricles. The bundle of His quickly divides into two main branches: the right bundle branch, which carries electrical impulses toward the right ventricle, and the left bundle branch, which conducts impulses toward the left ventricle. The left bundle branch further subdivides into anterior and posterior fascicles, allowing for more precise control of electrical activation throughout the larger and more complex left ventricle. These bundle branches consist of specialized cardiac cells that conduct electrical impulses much faster than ordinary heart muscle, ensuring that ventricular activation occurs rapidly and synchronously. The cells of the conduction system are larger than typical cardiac muscle cells and contain fewer contractile elements, as their primary function is electrical conduction rather than mechanical contraction. The final components of this electrical network are the Purkinje fibers, named after the Czech physiologist Johannes Purkinje, who first observed these distinctive cells. Purkinje fibers form an extensive network throughout both ventricles, branching repeatedly like the roots of a tree, to reach virtually every region of ventricular muscle. These fibers represent the most specialized conducting cells in the heart, capable of transmitting electrical impulses at speeds of up to 4 meters per second, roughly 4 times faster than ordinary cardiac muscle. The Purkinje fiber network ensures that electrical activation spreads rapidly throughout both ventricles, allowing for coordinated contraction that efficiently ejects blood from the heart. The fiber distribution is particularly dense in areas that need to activate early in the contraction sequence, such as the papillary muscles that control the mitral and tricuspid valves. This early activation ensures that these crucial structures are ready to prevent backward flow of blood as ventricular pressure rises. The propagation of electrical impulses through heart tissue follows principles similar to those governing electrical conduction in other biological systems, yet with important cardiac-specific adaptations. When an electrical impulse reaches a cardiac muscle cell, it causes voltage-gated sodium channels in the cell membrane to open rapidly, allowing sodium ions to rush into the cell and creating a rapid change in electrical charge called depolarization. This depolarization spreads to adjacent cells through gap junctions in the intercalated discs, creating a wave of electrical activity that travels throughout the heart muscle. However, cardiac cells have a unique feature called a plateau phase in their action potential, during which the cell remains depolarized for a relatively long period compared to other excitable tissues. This plateau phase, lasting 200 to 300 milliseconds, prevents the heart from contracting again until it has had time to relax and refill with blood. The transformation of electrical activity into mechanical contraction involves a sophisticated process called excitation-contraction coupling. When electrical depolarization reaches cardiac muscle cells, it triggers the release of calcium ions from internal storage sites within the cell. These calcium ions bind to regulatory proteins associated with the contractile machinery, allowing actin and myosin filaments to interact and generate force. The strength of cardiac contraction can be influenced by various factors that affect this excitation-contraction coupling process. Sympathetic nervous system stimulation, for example, increases the amount of calcium available for contraction, leading to stronger heartbeats. Certain medications can also influence calcium handling within cardiac cells, either enhancing or reducing the force of contraction. The heart's electrical system demonstrates remarkable adaptability, capable of adjusting both heart rate and conduction patterns in response to changing physiological demands. During exercise, sympathetic nervous stimulation increases the firing rate of the sinoatrial node and speeds conduction throughout the system, allowing for the faster heart rates needed to meet increased oxygen demands. Conversely, parasympathetic stimulation during rest slows AST node firing and can even temporarily block conduction through the AV node, creating the slower heart rates associated with relaxation and sleep. As your heart beats steadily in your chest right now, this intricate electrical network is orchestrating each contraction with millisecond precision, generating impulses, conducting them through specialized pathways, and coordinating the mechanical events that sustain your circulation through the elegant marriage of electricity and muscle contraction. The heart faces a unique circulatory challenge that sets it apart from every other organ in your body. While most organs can simply absorb oxygen and nutrients from the blood flowing through them, your heart muscle cannot sustain itself on the blood rushing through its chambers. Instead, this tireless organ requires its own dedicated blood supply system, a network of vessels so crucial that any interruption in their function can be immediately life-threatening. This coronary circulation represents one of the most elegant and essential vascular systems in your body, feeding the very muscle that keeps all other circulation flowing. The name coronary comes from the Latin word corona, meaning crown, because these vessels encircle the heart like a royal diadem. This poetic nomenclature reflects the critical importance of these vessels, as they truly crown the cardiovascular system with their life-sustaining function. The coronary circulation begins at the very source of the body's highest pressure blood flow, branching directly from the ascending aorta, just as oxygen-rich blood leaves the left ventricle. Two main coronary arteries arise from the aortic root, each emerging from small openings called coronary ostia, located just above the aortic valve cusps. The timing of this origin is no accident. These vessels receive blood at the peak pressure generated by left ventricular contraction, ensuring that the heart muscle receives oxygenated blood at the highest available pressure in the entire circulatory system. The right coronary artery emerges from the right aortic sinus and follows a path along the groove between the right atrium and right ventricle, known as the atrioventricular groove or coronary sulcus. This vessel typically measures 3 to 4 millimeters in diameter and supplies blood to several crucial regions of the heart. As it travels around the right side of the heart, the right coronary artery gives rise to several important branches, including the posterior descending artery and the acute marginal arteries. The territory supplied by the right coronary artery includes the right atrium, most of the right ventricle, the inferior wall of the left ventricle, and often the posterior portion of the interventricular septum. In many individuals, the right coronary artery also gives rise to the posterior left ventricular arteries, making it responsible for supplying blood to nearly half of the heart muscle. Additionally, the right coronary artery frequently supplies both the sinoatrial node and the atrioventricular node, making it crucial not only for heart muscle nutrition but also for the electrical conduction system that coordinates each heartbeat. The left coronary artery, often called the left main coronary artery, emerges from the left aortic sinus and follows a shorter but equally critical path. This vessel is typically larger than the right coronary artery, measuring 4 to 5 millimeters in diameter, reflecting the greater amount of heart muscle it must supply. After traveling only 1 to 2 centimeters, the left main coronary artery divides into two major branches that supply the larger and more muscular left side of the heart. The left anterior descending artery, often abbreviated as the LAD, travels down the front surface of the heart in a groove called the anterior interventricular sulcus. This vessel is sometimes called the Widowmaker by medical professionals, a grim nickname that reflects the potentially fatal consequences of blockage in this crucial artery. The LAD supplies the anterior wall of the left ventricle, the anterior portion of the interventricular septum, and often the apex of the heart. Its branches, called diagonal arteries and septal perforators, ensure that the thick muscular wall of the left ventricle receives adequate blood supply throughout its depth. The left circumflex artery follows the left atrioventricular groove, traveling around the left side of the heart, toward the back. This vessel supplies the lateral and posterior walls of the left ventricle through branches called obtuse marginal arteries. In some individuals, the circumflex artery also gives rise to the posterior descending artery, creating what cardiologists call a left dominant circulation pattern. The relative dominance of right versus left coronary systems varies among individuals, creating unique patterns of blood supply that can influence how heart disease manifests in different people. The distribution pattern of coronary arteries demonstrates remarkable individual variation, a fact that has important implications for both cardiac surgery and the treatment of coronary artery disease. While the basic pattern of right and left coronary systems remains consistent, the exact territory supplied by each vessel can vary significantly. Some people have additional vessels, such as a ramus intermedius artery that branches from the left main coronary artery, while others may have coronary arteries that follow unusual paths or supply atypical territories. After delivering oxygen and nutrients to the heart muscle, blood must be collected and returned to the heart through the coronary venous system. This network of veins follows pathways roughly parallel to the coronary arteries, but flows in the opposite direction. The coronary venous system is unique among venous networks because most of its blood does not return to the right atrium through the usual venous channels, but instead drains directly into the heart through specialized openings. The largest component of coronary venous drainage is the coronary sinus, a wide venous channel that runs along the posterior atrioventricular groove and opens directly into the right atrium. The coronary sinus receives blood from the great cardiac vein, which drains the anterior left ventricle, the middle cardiac vein, which drains the inferior wall, and the small cardiac vein, which drains portions of the right ventricle. Additional small veins, called Thebesian veins, drain directly into the cardiac chambers, particularly the right atrium and right ventricle, providing alternative drainage pathways for heart muscle blood flow. Beyond the coronary circulation lies the realm of the great vessels, the major arteries and veins that connect your heart to the rest of your circulatory system. These vessels represent the highways of circulation, carrying blood to and from your heart, with the capacity to handle the enormous volumes pumped with each heartbeat. The aorta stands as the body's main arterial highway, originating from the left ventricle and carrying oxygen-rich blood to every region of your body. This remarkable vessel begins as the ascending aorta, rising from the left ventricle, and giving rise to the coronary arteries before curving over the top of the heart to form the aortic arch. From the arch emerge the vessels that supply your head, neck and arms: the brachiocephalic trunk, left common carotid artery, and left subclavian artery. The aorta then descends through your chest and abdomen, giving rise to branches that supply your organs and tissues before finally dividing into the iliac arteries that supply your pelvis and legs. Throughout its course, the aorta must withstand the highest pressures in your circulatory system, while maintaining the flexibility to expand and contract with each heartbeat, storing energy during systole and releasing it during diastole to maintain continuous blood flow. The pulmonary vessels handle the crucial task of carrying blood between your heart and lungs for gas exchange. The pulmonary trunk emerges from the right ventricle, carrying oxygen-poor blood toward the lungs. This vessel quickly divides into right and left pulmonary arteries, each supplying one lung through an intricate network of branching vessels that parallel the airways. After gas exchange occurs in the lungs, 4 pulmonary veins typically return oxygen-rich blood to the left atrium, completing the pulmonary circulation loop. The vena cava represent the body's great venous return pathways, collecting oxygen-poor blood from throughout your body and delivering it back to the right atrium. The superior vena cava drains blood from your head, neck, arms and upper chest, while the inferior vena cava returns blood from your abdomen, pelvis and legs. These vessels must handle enormous volumes of blood while operating under much lower pressures than the arterial system, relying on one-way valves and the assistance of surrounding muscles to maintain flow back to the heart. One of the most remarkable features of coronary circulation is its capacity for adaptation through collateral circulation. When coronary arteries become narrowed or blocked, the heart can develop alternative pathways for blood flow through the growth of new vessels or the enlargement of existing small vessels. These collateral vessels can provide alternative routes for blood to reach heart muscle, sometimes preventing damage even when major coronary arteries become severely diseased. This adaptive capacity varies among individuals and can be enhanced through regular exercise, which stimulates the development of these protective alternative pathways. As your heart beats steadily in your chest, this intricate network of vessels is working continuously to ensure that every cell of heart muscle receives the oxygen and nutrients it needs to sustain its tireless contractions, while the great vessels carry the life-giving blood your heart pumps to every corner of your body. Flowing through your circulatory system at this very moment is a remarkable fluid that serves as your body's river of life, carrying precious cargo to every cell while whisking away the waste products of cellular metabolism. Blood represents far more than a simple transport medium. It is a complex living tissue that performs dozens of essential functions, from delivering oxygen to fighting infections to maintaining the delicate chemical balance that keeps you alive. This crimson fluid, comprising roughly 8% of your body weight, travels through 60,000 miles of vessels, completing its circuit through your entire body in less than 1 minute. Blood is not the uniform red liquid it might appear to be at first glance, but rather a sophisticated mixture of plasma, cells and countless dissolved substances, each component playing crucial roles in maintaining life. When blood is allowed to settle, or is separated by centrifugation, it reveals its true complexity, separating into distinct layers that tell the story of its composition. The bottom layer, comprising about 45% of blood volume, consists of red blood cells packed tightly together. Above this lies a thin, pale layer called the buffy coat, containing white blood cells and platelets. The top layer, making up roughly 55% of blood volume, is plasma, the liquid component that serves as the transport medium for all other blood elements. Plasma itself is a remarkable substance, consisting of about 90% water mixed with an intricate array of dissolved proteins, nutrients, hormones, gases, and waste products. This pale yellow fluid serves as your body's internal ocean, maintaining the proper chemical environment for cellular function while transporting substances between distant parts of your body. The proteins dissolved in plasma play particularly crucial roles, including albumin, which maintains proper fluid balance between your blood vessels and tissues, and an array of clotting factors that prevent excessive bleeding when blood vessels are damaged. Floating within this plasma ocean are the red blood cells, perhaps the most specialized and numerous cells in your entire body, also called erythrocytes. These cells number approximately 25 trillion in an average adult, with each cubic millimeter of blood containing about 5 million of these oxygen-carrying specialists. Red blood cells possess a unique biconcave disc shape, resembling tiny donuts with the centers pushed in rather than punched out. This distinctive shape maximizes surface area for gas exchange, while allowing the cells to squeeze through capillaries narrower than their own diameter. The remarkable efficiency of red blood cells in carrying oxygen stems from their unusual cellular structure and composition. Mature red blood cells have sacrificed their nuclei and most organelles to make room for millions of hemoglobin molecules, the iron-containing proteins responsible for oxygen transport. Each red blood cell contains approximately 280 million hemoglobin molecules, and each hemoglobin molecule can carry 4 oxygen atoms, giving each tiny cell the capacity to transport over 1 billion oxygen atoms throughout your body. Hemoglobin's oxygen-carrying ability depends on iron atoms at the center of each hemoglobin molecule, which can reversibly bind to oxygen molecules. In the high oxygen environment of your lungs, hemoglobin eagerly binds oxygen, becoming bright red oxyhemoglobin. As blood travels through tissues where oxygen concentrations are lower, hemoglobin releases its oxygen cargo, allowing it to diffuse into cells that need it for energy production. The hemoglobin then picks up carbon dioxide, a waste product of cellular metabolism, and carries it back to the lungs for elimination. The lifespan of red blood cells reflects both their specialized function and their structural limitation. These cells circulate through your bloodstream for approximately 120 days, traveling hundreds of miles through increasingly small vessels while maintaining their flexibility and function. However, the absence of a nucleus means they cannot repair themselves or produce new proteins, and they gradually become less flexible and more fragile. Eventually, specialized cells in your spleen and liver recognize these aging red blood cells and remove them from circulation, recycling their components to create new blood cells. Sharing the bloodstream with red blood cells are the white blood cells, or leukocytes, your body's mobile immune system. While far less numerous than red blood cells, with only about 7,000 white blood cells per cubic millimeter of blood, these cells play absolutely critical roles in defending against infection and disease. White blood cells come in several distinct varieties, each is specialized for different aspects of immune defense. Neutrophils represent the most abundant type of white blood cell, serving as your body's first responders to bacterial infections. These cells can quickly migrate from blood vessels into tissues, where they engulf and destroy bacteria through a process called phagocytosis. Lymphocytes, including B cells and T cells, coordinate more sophisticated immune responses, with B cells producing antibodies and T cells directly attacking infected or abnormal cells. Monocytes patrol the bloodstream and can transform into macrophages, large cells that consume cellular debris and coordinate immune responses in tissues. The third major cellular component of blood consists of platelets, tiny cell fragments that play essential roles in preventing blood loss when blood vessels are damaged. Platelets, also called thrombocytes, are not complete cells, but rather fragments of large cells called megakaryocytes that reside in your bone marrow. Each platelet measures only 2 to 3 micrometers in diameter, making them much smaller than red or white blood cells, but they circulate in enormous numbers, typically 200,000 to 400,000 per cubic millimeter of blood. When blood vessel damage occurs, platelets demonstrate remarkable responsiveness, adhering to the damaged vessel wall within seconds, and releasing chemical signals that attract more platelets to the site. These activated platelets change shape, becoming spiny and sticky, allowing them to clump together and form a temporary plug that helps stop bleeding. Simultaneously, platelets release chemicals that initiate the complex cascade of reactions, leading to blood clot formation. The clotting process itself represents one of the most sophisticated biochemical cascades in your body, involving dozens of proteins that work together to convert liquid blood into a solid clot when and where it's needed. This process must be precisely controlled, as too little clotting leads to excessive bleeding, while too much clotting can block blood vessels and cause heart attacks or strokes. The balance between clot formation and clot dissolution is maintained through intricate feedback mechanisms that ensure clots form only when truly needed and dissolve when healing is complete. The volume of blood in your circulatory system, typically 5 to 6 liters in an average adult, represents a carefully regulated quantity that must be maintained within narrow limits for optimal function. This blood volume creates the pressure necessary to drive circulation through your vast network of blood vessels, from the largest arteries down to the tiniest capillaries. The distribution of this blood volume changes constantly as your body responds to different demands and conditions. At any given moment, roughly 20% of your blood volume circulates through your arteries, creating the high-pressure system that drives blood flow to your tissues. About 5% flows through capillaries, where the actual exchange of oxygen, nutrients, and waste products occurs between blood and tissues. The remaining 75% resides in your venous system, serving as a reservoir that can be mobilized when additional blood flow is needed. The journey of blood from your heart to your tissues and back again represents a remarkable feat of biological engineering. Oxygen-rich blood leaves your left ventricle under high pressure, traveling through progressively smaller arteries until it reaches the vast network of capillaries where oxygen and nutrients transfer to tissues. The now oxygen-poor blood begins its return journey through increasingly larger veins, ultimately reaching your right atrium and completing the systemic circuit. Simultaneously, a separate circuit carries oxygen-poor blood from your right ventricle to your lungs, where it releases carbon dioxide and picks up fresh oxygen before returning to your left atrium. As your heart beats quietly in your chest right now, this precious river of life is flowing continuously through your body, delivering the oxygen and nutrients that sustain every cell, while removing the waste products that could harm you, maintaining the delicate balance of chemistry and pressure that keeps you alive with every beat. Every heartbeat represents a precisely choreographed sequence of events that transforms your heart from a relaxed, blood-filled chamber into a powerful pump and back again, all within the span of less than 1 second. This rhythmic sequence, known as the cardiac cycle, occurs roughly 70 times per minute throughout your entire life. Yet, each cycle unfolds with such mechanical precision that it rivals the finest clockwork. Understanding this cycle reveals how your heart coordinates electrical signals, pressure changes, valve movements, and blood flow to create the steady pulse that sustains your existence. The cardiac cycle traditionally divides into 2 main phases that reflect the fundamental activities of your heart muscle. Systole represents the contraction phase, when your heart muscle squeezes forcefully to eject blood into your arteries. Diastole encompasses the relaxation phase, when your heart muscle relaxes and allows its chambers to refill with blood for the next contraction. However, this apparently simple division masks a complex sequence of precisely timed events that ensure blood flows efficiently in only one direction through your circulatory system. Systole begins when electrical impulses from your heart's conduction system trigger the coordinated contraction of ventricular muscle fibers. This phase represents the heart's active work period, when mechanical energy is generated and blood is forcefully ejected from the ventricles. Systole can be further subdivided into 2 distinct periods: isovolumic contraction and ventricular ejection, each characterized by specific pressure relationships and valve positions. During isovolumic contraction, ventricular muscle begins contracting while all 4 heart valves remain closed. The term isovolumic means same volume, reflecting the fact that no blood enters or leaves the ventricles during this brief period. As ventricular pressure rises rapidly due to muscle contraction, it soon exceeds the pressure in the relaxed atria, causing the tricuspid and mitral valves to snap shut with an audible sound. However, ventricular pressure has not yet risen high enough to overcome the pressure in the aorta and pulmonary arteries, so the semi-lunar valves remain closed as well. This phase creates a brief moment when the ventricles are completely sealed chambers, with contracting muscle causing pressure to rise dramatically without any change in blood volume. The pressure increase during isovolumic contraction is remarkably steep, rising from near 0 to over 80 millimeters of mercury in the left ventricle within just 50 milliseconds. This rapid pressure rise demonstrates the explosive force generated when cardiac muscle contracts in unison. Ventricular ejection begins when rising ventricular pressure finally exceeds the pressure in the great arteries, forcing the aortic and pulmonary valves to open suddenly. Blood rushes out of the ventricles with considerable velocity, creating the stroke volume that will carry oxygen and nutrients throughout your body. During this ejection phase, ventricular pressure continues to rise initially, reaching peak values of 120 millimeters of mercury or higher in the left ventricle, before gradually declining as the ventricles empty. The ejection phase demonstrates the remarkable efficiency of cardiac design. Despite the ventricles never emptying completely, they typically eject 60 to 70% of their blood volume with each contraction. This ejection fraction represents a crucial measure of cardiac function, as it reflects how effectively the heart converts its muscular contraction into useful blood flow. Diastole represents the heart's relaxation and recovery period, when ventricular muscle relaxes and chambers refill with blood in preparation for the next contraction. Like systole, diastole consists of distinct phases characterized by specific valve positions and pressure relationships. The transition from systole to diastole begins when ventricular muscle starts to relax, causing ventricular pressure to fall rapidly. Isovolumic relaxation marks the beginning of diastole, occurring when ventricular pressure falls below arterial pressure, causing the aortic and pulmonary valves to close with another audible sound. During this phase, all 4 heart valves are again closed, but now the ventricles are relaxing rather than contracting. Ventricular pressure continues to fall precipitously as muscle fibers lengthen and the chambers begin to return to their resting shape. Ventricular filling begins when ventricular pressure falls below atrial pressure, causing the tricuspid and mitral valves to open and allowing blood to flow from atria to ventricles. This filling process occurs in 2 distinct phases: passive filling and active filling. During passive filling, blood flows from the higher pressure atria into the lower pressure ventricles, simply due to pressure differences, with no muscular assistance required. Active filling occurs near the end of diastole when the atria contract, providing an additional push that tops off ventricular filling just before the next heartbeat begins. This atrial contraction, sometimes called the atrial kick, typically contributes about 20 to 30% of ventricular filling, though the heart can function adequately without it if atrial contraction is impaired. The timing relationship between atrial and ventricular contractions reveals the sophisticated coordination built into the cardiac cycle. Atrial systole occurs during ventricular diastole, ensuring that atria contract when ventricles are relaxed and ready to receive blood. Conversely, ventricular systole occurs during atrial diastole, allowing atria to fill while ventricles are ejecting blood. This alternating pattern maximizes the efficiency of blood flow through the heart. Pressure changes throughout the cardiac cycle create a dynamic interplay between different chambers and vessels that drives circulation forward. Left ventricular pressure varies from near zero during diastole to over 120 millimeters of mercury during systole, creating the pressure gradient necessary to drive blood through the systemic circulation. Right ventricular pressures are much lower, typically reaching only 25 millimeters of mercury during systole, reflecting the lower resistance of pulmonary circulation. These pressure changes directly control valve function throughout the cardiac cycle. Heart valves operate purely as passive structures, opening when pressure is higher on their inflow side, and closing when pressure becomes higher on their outflow side. The precise timing of valve opening and closing creates the characteristic heart sounds that can be heard with a stethoscope. The familiar lub-dub rhythm of your heartbeat originates from the closing of heart valves during specific phases of the cardiac cycle. The first heart sound, lub, occurs at the beginning of systole, when the tricuspid and mitral valves close as ventricular pressure rises above atrial pressure. The second heart sound, dub, occurs at the beginning of diastole, when the aortic and pulmonary valves close as ventricular pressure falls below arterial pressure. The coordination between electrical and mechanical events represents perhaps the most remarkable aspect of the cardiac cycle. Each mechanical event follows its corresponding electrical event by a precise interval, demonstrating how the heart's conduction system controls the timing of mechanical contraction. The electrical depolarization of ventricles precedes mechanical contraction by about 50 milliseconds, while electrical repolarization occurs during mechanical relaxation. As your heart beats steadily in your chest right now, this intricate cardiac cycle is repeating with metronomic precision, coordinating electrical signals, pressure changes, valve movements, and muscle contractions to create the rhythmic flow that sustains your life with every beat. The story of your heart begins long before your first breath, in the earliest weeks of embryonic development when you were no larger than a grain of rice. During the 3rd week after conception, when most mothers don't yet know they're pregnant, a remarkable transformation begins, as simple clusters of cells start organizing themselves into the structure that will become your heart. This embryonic heart formation represents one of the most critical developmental processes in human biology, as any disruption during these crucial early weeks can have lifelong consequences. The heart is actually the first organ to function in the developing embryo, beginning its rhythmic contractions around day 22 of gestation. At this point, your future heart consists of nothing more than a simple tube, yet it already demonstrates the fundamental property that will define it throughout your life: the ability to contract spontaneously and rhythmically. This primitive heart tube begins folding and twisting in a precise sequence, gradually developing the loops and chambers that will eventually become the 4-chambered heart you possess today. During the 4th and 5th weeks of embryonic development, the simple heart tube undergoes a process called cardiac looping, where it folds back on itself in a characteristic S-shaped curve. This looping process is essential for proper heart development, as it brings the future atria and ventricles into their correct anatomical positions. The precision required for this process is extraordinary. The heart must loop in exactly the right direction, and to exactly the right degree, or serious developmental maladies will result. By the 7th week of development, your embryonic heart has developed all 4 chambers, though they are still quite different from their mature counterparts. The walls are thin, the valves are just beginning to form, and the electrical conduction system is still developing. However, this tiny heart, no larger than a poppy seed, is already pumping blood through a rapidly developing circulatory system, sustaining the growth of your embryonic body. Fetal circulation represents one of the most ingenious adaptations in human development, solving the challenge of how to circulate blood through a developing body when the lungs are not yet functioning. Since the fetus receives oxygen from the mother's blood through the placenta rather than from its own lungs, fetal circulation includes several special pathways that bypass the non-functioning lungs and redirect blood flow to where it's needed most. The most remarkable feature of fetal circulation is the presence of 3 temporary structures that allow blood to bypass the lungs and flow directly to the body's organs. The ductus arteriosus connects the pulmonary artery directly to the aorta, allowing blood pumped by the right ventricle to bypass the lungs and flow directly into systemic circulation. The foramen ovale, an opening in the wall between the left and right atria, allows blood to flow directly from the right atrium to the left atrium, bypassing the right ventricle and lungs entirely. The ductus venosus allows blood from the placenta to bypass the developing liver and flow directly to the heart. These fetal circulatory adaptations create a system where the right and left sides of the heart work in parallel rather than in series, with both ventricles pumping blood to the body rather than the left ventricle handling systemic circulation and the right ventricle handling pulmonary circulation. This arrangement ensures that the most oxygen-rich blood from the placenta is directed to the brain and other vital organs, while less oxygenated blood supplies the lower body. The transition at birth represents one of the most dramatic physiological changes that occurs during human life, as your circulatory system must transform from the fetal pattern to the adult pattern within minutes of your first breath. When you took your first breath, the expansion of your lungs created a dramatic decrease in pulmonary vascular resistance, suddenly making it easier for blood to flow through the lungs than through the systemic circulation. This change in resistance patterns immediately alters the pressure relationships within your heart, causing the foramen ovale to close, as left atrial pressure now exceeds right atrial pressure. The ductus arteriosus begins constricting within hours of birth, as oxygen levels rise and certain hormones decrease, gradually closing this pathway between the pulmonary artery and aorta. The ductus venosus closes as blood flow from the placenta ceases, and the liver begins handling all blood returning from the lower body. These changes at birth represent a one-way transformation that cannot be reversed. Once the fetal circulatory pathways close, your heart has committed to the adult circulation pattern that will serve you for the rest of your life. The remarkable precision of this transition explains why the first few hours and days after birth are such a critical period, as the circulatory system must successfully complete this complex reorganization. Heart growth through childhood and adolescence follows patterns that reflect both the increasing size of your body and the changing demands placed on your cardiovascular system. During the first year of life, your heart doubles in weight as it adapts to the increased workload of postnatal circulation. Throughout childhood, heart size increases roughly in proportion to body size, maintaining the relationship between cardiac output and the metabolic needs of your growing tissues. Adolescence brings unique challenges to the cardiovascular system, as rapid growth spurts can temporarily create situations where heart size lags behind body size, sometimes leading to the feeling of being easily winded during periods of rapid growth. However, the heart demonstrates remarkable adaptability during this period, adjusting its size and function to meet the changing demands of the maturing body. Adult heart maintenance represents a delicate balance between preserving function and adapting to changing demands throughout the decades of adult life. Unlike many organs that can regenerate damaged cells, the heart has very limited regenerative capacity, making the preservation of existing cardiac muscle cells crucial for lifelong function. The adult heart maintains itself through efficient energy utilization, careful regulation of calcium handling within cardiac cells, and the continuous renewal of cellular components even though the cells themselves are not replaced. The heart's ability to adapt to changing demands throughout adult life demonstrates remarkable plasticity. Regular exercise can increase heart muscle mass and improve efficiency, While prolonged inactivity can lead to deconditioning and decreased cardiac reserve, the heart adapts to pregnancy by increasing output to support both mother and developing fetus, and it adjusts to the decreased demands of advanced age by maintaining function despite gradual changes in muscle properties. Age-related changes in heart function represent a natural part of the aging process, though the extent of these changes varies considerably among individuals. The heart muscle gradually becomes less flexible. The electrical conduction system may slow slightly, and the maximum heart rate achievable during exercise decreases predictably with age. However, a healthy heart retains remarkable reserve capacity even into advanced age, maintaining adequate function for normal activities throughout a typical human lifespan. As your heart beats quietly in your chest right now, it continues the same fundamental rhythm that began before your birth, adapted through the dramatic changes of early development, and will continue faithfully until your final breath, representing the most enduring rhythm of your entire existence. Your heart possesses one of the most remarkable adaptive capacities in the human body, capable of adjusting its function across a vast range of demands while maintaining the steady reliability that sustains your life. This adaptability extends far beyond simply beating faster or slower. Your heart can modify its size, strength, efficiency, and coordination in response to everything from physical exercise to emotional stress, from changes in altitude to the natural rhythms of sleep and wakefulness. Understanding these adaptations reveals how your heart serves not just as a mechanical pump, but as a dynamic organ that continuously fine-tunes itself to meet your body's ever-changing needs. Exercise represents perhaps the most dramatic example of cardiac adaptation, transforming the heart in ways that enhance its efficiency and reserve capacity. When you engage in regular physical activity, your heart responds by becoming larger, stronger, and more efficient, much like any muscle that is regularly challenged. However, cardiac adaptations to exercise extend far beyond simple enlargement, involving sophisticated changes in heart muscle properties, electrical conduction, and circulatory efficiency. Aerobic exercise, such as running, cycling, or swimming, primarily promotes what cardiologists call eccentric cardiac hypertrophy, where heart chambers enlarge while maintaining or even slightly decreasing wall thickness. This adaptation allows the heart to hold more blood and pump larger volumes with each beat, increasing stroke volume and reducing the heart rate required to maintain any given level of cardiac output. Athletes who engage in regular endurance training often develop resting heart rates in the 40s or 50s, not because their hearts are weak, but because each beat pumps so much blood that fewer beats are needed to maintain circulation. Resistance exercise and strength training promote a different pattern of cardiac adaptation called concentric hypertrophy, where heart muscle walls become thicker and more powerful without necessarily increasing chamber size. This adaptation enhances the heart's ability to generate high pressures and maintain circulation during activities that involve sustained muscle contractions and temporary increases in blood pressure. The athletic heart demonstrates remarkable efficiency compared to untrained hearts, requiring less oxygen for its own function while generating greater cardiac output when needed. Trained hearts also develop enhanced electrical stability, with improved coordination between different regions of heart muscle and more consistent conduction of electrical impulses. These electrical improvements contribute to the enhanced performance and reduced risk of rhythm disturbances often observed in well-trained athletes. Altitude presents your heart with a unique set of challenges that trigger specific adaptive responses designed to maintain adequate oxygen delivery, despite reduced atmospheric pressure. When you travel to high altitude, the decreased partial pressure of oxygen in the air means that each breath delivers less oxygen to your lungs, forcing your cardiovascular system to work harder to meet your body's oxygen needs. The immediate response to altitude involves an increase in heart rate and cardiac output, as your heart attempts to circulate blood faster to compensate for its reduced oxygen content. Within hours of altitude exposure, your heart rate may increase by 20 to 30 beats per minute even at rest, a response mediated by chemoreceptors that detect the decrease in blood oxygen levels and trigger sympathetic nervous system activation. Over days and weeks at altitude, more sophisticated adaptations develop that improve oxygen delivery efficiency. Your body produces more red blood cells, increasing the oxygen carrying capacity of your blood and reducing the workload on your heart. Your heart muscle itself may undergo subtle adaptations that improve its efficiency in extracting oxygen from blood, while changes in peripheral circulation help ensure that available oxygen reaches vital organs despite the challenging conditions. Stress responses reveal another fascinating dimension of cardiac adaptability. as your heart responds not only to physical demands, but also to psychological and emotional challenges. When you encounter stress, whether physical danger or emotional pressure, your sympathetic nervous system releases hormones like epinephrine and norepinephrine that dramatically alter cardiac function within seconds. These stress hormones increase heart rate, strengthen heart muscle contractions, and enhance the heart's electrical conduction velocity, preparing your cardiovascular system for the increased demands that might accompany a fight or flight response. However, your heart's response to stress involves more than just beating faster and harder. It includes subtle changes in heart rate variability, the natural fluctuation in time intervals between heartbeats that reflects the balance between sympathetic and parasympathetic nervous system influences. Heart rate variability serves as a window into your autonomic nervous system function and overall cardiovascular health. Healthy hearts demonstrate significant variability between beats, with this variability decreasing under stress and increasing during relaxation and recovery. This variability reflects your heart's ability to respond appropriately to changing demands, with higher variability generally indicating better cardiovascular fitness and stress resilience. Sleep and circadian rhythms exert profound influences on cardiac function, with your heart following natural daily patterns that optimize its performance for different phases of your 24-hour cycle. During sleep, your heart rate naturally decreases, blood pressure drops, and heart rate variability increases, allowing your cardiovascular system to rest and recover from the day's demands. The transition from wakefulness to sleep involves a shift from sympathetic to parasympathetic nervous system dominance, creating the slower, more variable heart rhythms characteristic of healthy sleep. Deep sleep stages are associated with the most dramatic reductions in heart rate and blood pressure, providing crucial recovery time for your cardiovascular system. Circadian rhythms also influence the timing of cardiac events, with heart attacks and strokes showing peak incidence in the early morning hours when blood pressure and heart rate naturally rise to prepare your body for awakening. Understanding these circadian patterns has led to improved timing of cardiac medications and procedures to work with rather than against your heart's natural rhythms. Emotional states demonstrate yet another dimension of cardiac adaptability, as feelings like joy, fear, anger, and sadness all produce characteristic changes in heart function. The phrase broken heart syndrome reflects a real medical condition, where intense emotional stress can temporarily impair heart muscle function, while positive emotions like love and contentment are associated with more stable heart rhythms and improved cardiovascular health. The connection between emotions and cardiac function involves complex interactions between your brain, nervous system, and heart, mediated by hormones, neurotransmitters, and direct neural connections. These emotional influences on heart function explain why psychological stress management and emotional well-being play such important roles in maintaining cardiovascular health throughout life. Perhaps most remarkable is your heart's reserve capacity, its ability to increase output far beyond resting levels when circumstances demand. A healthy heart can increase its output by 4 to 5 times above resting levels, pumping from the typical 5 liters per minute at rest to 25 liters per minute or more during maximum exercise. This enormous reserve ensures that your heart can meet the most extreme demands while maintaining adequate circulation to vital organs. As your heart beats steadily in your chest right now, it stands ready to adapt to whatever challenges await, possessing the remarkable flexibility to adjust its function across an enormous range of conditions while maintaining the steady reliability that sustains your life. The rhythm of your heart, while remarkably consistent in its basic pattern, displays a fascinating array of natural variations that reflect both your individual characteristics and your body's constant adaptation to changing conditions. These variations in heart function are not signs of malfunction, but rather evidence of your cardiovascular system's sophisticated ability to adjust its performance across different circumstances, ages, and individual needs. Understanding these natural rhythms and variations reveals how your heart maintains its essential function while accommodating the unique demands of your particular body and lifestyle. Heart rate represents the most obvious variable in cardiac function, yet what constitutes normal varies dramatically across different stages of life. A newborn infant's heart beats at a rapid pace of 120 to 160 beats per minute, reflecting the high metabolic demands of rapid growth and development. This quick rhythm gradually slows as children mature, with toddlers typically maintaining heart rates between 90 and 140 beats per minute, while school-age children settle into rates between 70 and 110 beats per minute. Adult heart rates typically range from 60 to 100 beats per minute at rest, though this broad range encompasses considerable individual variation based on fitness level, genetics, and overall health. Well-trained athletes often maintain resting heart rates in the 40s or 50s, not due to weakness but because their enlarged, efficient hearts pump more blood with each beat, requiring fewer contractions to maintain adequate circulation. Conversely, elderly adults may experience slight increases in resting heart rate as aging affects the heart's electrical conduction system and overall efficiency. Beyond simple heart rate lies the more subtle, but equally important concept of heart rate variability, the natural fluctuation in time intervals between individual heartbeats. A healthy heart does not beat with metronomic regularity. Instead, it displays constant minor variations in the timing between beats, reflecting the dynamic balance between sympathetic and parasympathetic nervous system influences on cardiac function. Heart rate variability serves as a window into your autonomic nervous system, health, and your heart's ability to respond appropriately to changing demands. Higher heart rate variability generally indicates better cardiovascular fitness, stress resilience, and overall health, while reduced variability may signal excessive stress, fatigue, or underlying health issues. This variability is most pronounced during sleep and relaxation, when parasympathetic nervous system activity dominates, and decreases during stress, exercise, or illness, when sympathetic activation takes precedence. One of the most beautiful examples of natural heart rate variation occurs with breathing, creating a phenomenon called respiratory sinus arrhythmia. As you breathe in, your heart rate naturally increases slightly, and as you exhale, it decreases. This synchronization between breathing and heart rate reflects sophisticated coordination between your respiratory and cardiovascular systems, optimizing the efficiency of oxygen uptake and delivery. During inspiration, the venous return to your heart increases as the diaphragm moves downward and creates negative pressure in the chest cavity, drawing more blood back to the right ventricle. Your heart responds by beating slightly faster to accommodate this increased blood flow. During expiration, venous return decreases and heart rate slows correspondingly. This breathing heart rate synchronization is most apparent during slow, deep breathing and represents a sign of healthy cardiovascular function. Individual differences in cardiac anatomy create another layer of variation in heart function, with each person's heart displaying unique characteristics that influence its performance. Heart size varies considerably among individuals, not just in proportion to body size but also in shape, chamber proportions, and wall thickness. Some people naturally have larger hearts that pump more blood per beat, while others have smaller but more rapidly beating hearts that achieve similar cardiac outputs through different mechanisms. The position of your heart within your chest varies among individuals, with some hearts sitting more vertically and others more horizontally. Tall, thin individuals often have hearts that hang more vertically within the chest cavity, while shorter, broader individuals typically have hearts positioned more horizontally. These positional differences affect how the heart appears on chest X-rays and electrocardiograms, but have no impact on function when they fall within normal ranges. Gender differences in heart structure and function represent another important source of variation in cardiac characteristics. Women typically have smaller hearts than men, even when accounting for differences in body size with correspondingly smaller coronary arteries and chamber dimensions. However, women's hearts often demonstrate superior efficiency per unit of muscle mass, and they typically maintain better electrical stability throughout their reproductive years. Women's hearts also respond differently to hormonal influences throughout the menstrual cycle, pregnancy, and menopause, with estrogen generally providing protective effects on cardiovascular function. These hormonal influences affect heart rate variability, blood vessel function, and overall cardiovascular risk patterns, creating gender-specific considerations in heart health management. Temperature changes elicit predictable responses from your heart, as it helps maintain your body's thermal balance. In hot conditions, your heart rate increases to support enhanced blood flow to your skin for cooling, while blood vessels near the surface dilate to promote heat loss. Cold exposure produces the opposite response, with heart rate initially increasing to maintain circulation as blood vessels constrict to preserve core body temperature. Positional changes also influence heart function, with your heart adapting quickly as you move from lying to sitting to standing positions, When you stand up, gravity pulls blood toward your legs, temporarily reducing the amount returning to your heart. Your cardiovascular system responds within seconds by increasing heart rate and constricting blood vessels to maintain adequate blood pressure and circulation to your brain. As your heart beats steadily in your chest right now, it embodies all these natural variations and adaptations, maintaining its essential rhythm while constantly fine-tuning its performance to meet your individual needs and circumstances. Your heart represents one of the most resilient and adaptable organs in your body, capable of maintaining its essential function across decades of life when supported by conditions that honor its biological needs and natural rhythms. The factors that support optimal heart function extend far beyond simple medical interventions, encompassing the fundamental lifestyle patterns, environmental conditions, and bodily relationships that allow this remarkable organ to express its full potential for strength, efficiency, and longevity. The foundation of heart health lies in regular physical activity that challenges your cardiovascular system appropriately and consistently. When you engage in activities that elevate your heart rate and increase the demands on your circulatory system, your heart responds by becoming stronger, more efficient, and better able to handle the stresses of daily life. This adaptation occurs not through occasional intense efforts but through the cumulative effect of regular moderate activity that allows your heart to develop its capacity gradually and sustainably. Movement serves your heart in multiple ways beyond simple strength building. Physical activity helps maintain the flexibility of your blood vessels, supports the development of collateral circulation that provides backup pathways for blood flow, and promotes the efficient function of your heart's electrical conduction system. Even gentle activities like walking or gardening provide benefits, as they encourage regular changes in heart rate and blood flow patterns that keep your cardiovascular system responsive and adaptable. The relationship between your heart and other organ systems reveals the interconnected nature of optimal health, demonstrating how supporting one system inevitably benefits others. Your respiratory system works in intimate partnership with your heart, with healthy lung function reducing the workload on your right ventricle and ensuring efficient oxygen exchange. Deep, regular breathing not only delivers oxygen to your bloodstream, but also assists venous return to your heart through the pumping action of your diaphragm. Your digestive system influences heart health through its role in processing nutrients that support cardiac muscle function and maintaining blood chemistry within optimal ranges. A well-functioning digestive system efficiently absorbs the minerals, vitamins, and other nutrients your heart needs for cellular energy production, electrical conduction, and tissue maintenance. The timing and composition of meals can influence heart rhythm and blood pressure patterns, with regular, balanced nutrition supporting steady cardiovascular function. Your nervous system maintains constant communication with your heart through both conscious and unconscious pathways, adjusting cardiac function in response to physical demands, emotional states, and environmental conditions. A healthy nervous system provides appropriate autonomic regulation that allows your heart rate and rhythm to vary naturally throughout the day while maintaining overall stability and efficiency. Signs of healthy heart function often manifest as patterns that reflect your heart's natural rhythms and adaptability rather than absolute measurements or fixed values. A healthy heart demonstrates appropriate responsiveness, increasing its rate and output when demands rise and settling into slower, more variable rhythms during rest and sleep. This responsiveness indicates that your heart's electrical and muscular systems are working in harmony and can adapt appropriately to changing conditions. Heart rate variability serves as one of the most revealing indicators of cardiovascular health, with a healthy heart showing natural fluctuations between beats that reflect balanced autonomic nervous system function. These subtle variations, barely perceptible without specialized equipment, indicate that your heart is neither rigidly fixed in its rhythm nor chaotically irregular, but rather demonstrates the flexible stability characteristic of optimal biological function. Your heart's incredible endurance capacity becomes apparent when you consider that this organ beats approximately 2.5 billion times over an average human lifespan, pumping roughly 50 million gallons of blood, while maintaining remarkable consistency in its performance. This endurance reflects not just the durability of cardiac muscle, but the efficiency of cellular energy systems, the effectiveness of repair mechanisms, and the organ's ability to adapt to changing demands while preserving its essential function. Daily rhythms reveal another dimension of heart health, as your cardiovascular system naturally follows circadian patterns that coordinate with your sleep-wake cycle, hormonal fluctuations, and metabolic demands. A healthy heart shows predictable daily variations, with lower heart rates and blood pressures during sleep, gradual increases upon awakening, and appropriate responses to the activities and stresses of waking hours. The rest periods your heart experiences during sleep represent crucial recovery time. When cellular repair processes are most active, stress hormone levels are lowest, and your cardiovascular system can restore its reserves for the next day's demands. Quality sleep supports heart health, not just through this direct rest effect, but also through its influence on hormonal balance, stress resilience, and overall physiological harmony. Perhaps the most profound aspect of heart health lies in developing what might be called cardiac awareness, a gentle attentiveness to the messages your heart continuously provides about your overall state of well-being. This awareness doesn't involve anxious monitoring or obsessive checking, but rather a quiet sensitivity to the natural rhythms and responses that indicate when your heart is functioning harmoniously within your body's larger systems. As your heart beats quietly and steadily in your chest right now, it embodies the remarkable potential for sustained health and vitality that emerges when biological systems are supported by appropriate conditions and allowed to express their inherent wisdom and resilience. As our gentle exploration of the human heart draws to a close, take a moment to simply listen. There it is, that steady, faithful rhythm that has accompanied us throughout this journey. Your heart, beating quietly in your chest right now, represents billions of years of evolutionary refinement, millions of synchronized cellular activities, and the simple, profound miracle of sustained life. Throughout these chapters we've discovered chambers and valves, electrical networks and muscular walls, vessels that feed the heart and rhythms that never cease. Yet perhaps the most remarkable aspect of your heart is not any single component, but rather how seamlessly all these elements work together in perfect harmony. Your heart asks nothing of your conscious mind, requires no deliberate effort, makes no demands for your attention. It simply works, beat after beat, sustaining the quiet miracle of your existence. Consider that your heart began its rhythmic contractions when you were no larger than a poppy seed, and it will continue its dedicated service throughout every moment of your life. It adapts to your needs, responds to your emotions, and provides the fundamental rhythm upon which all other biological processes depend. As you settle into rest, let the knowledge of your heart's remarkable design bring you comfort. This organ, no larger than your fist, embodies millions of years of biological wisdom, knowing precisely how to maintain the delicate balance that sustains life, with trustworthy, quiet competence through every beat of your peaceful night.